Welcome to the session on the anatomy of the brain. The human brain is extraordinarily complex. The best estimate suggests that it contains between 10 and 100 billion cells connecting into rich networks. These connections develop as we live our lives. Our actions, our emotions, our thinking, our behaviour are all governed by the brain. The wide range of human personalities and activities reflects the way in which our brains have developed. Physical damage or mental trauma can result in behaviour that lies outside the norm. In this activity, you will have the opportunity to explore brain anatomy, to learn about the links between anatomy and behaviour, and to consider the neuroanatomical basis of autism. The overall shape of the brain. Brains are broadly similar. There are two hemispheres, each covered by the deeply folded cortex. The cortex is sometimes known as the grey matter. The colour derives from the presence of the cell bodies. The cortical folds create solci, or the valleys as it's known, the gyri that forms the crowns between the valleys. Although the detail of these folds vary, they are prominent folds that are found in most people. They create the familiar brain shape. Underlying the cortex is the white matter. It contains the nerve tracts that link the cells of the cortex. Let's explore the lobes. We covered aspects of this on day one. Think about the discussions we had around the brain imaging scans and how they're used to assess for structural damage and the consequential diagnosis of autistic spectrum conditions. Each hemisphere is divided into 4 lobes, each of which is specialist functions while working with other lobes to carry out complex processing tasks. The frontal lobe is the part of the brain that initiates actions and activity. It underpins reasoning, planning, problem solving, articulation of speech, movement, and the generation of emotional states, and influences activity in all other lobes. The parietal lobe receives stimuli related to touch, pressure, temperature and pain. The area has the capacity to integrate information from both new stimuli and from memory. It acts as a clearinghouse. The temporal lobe receives input from the ears and processes these stimuli to generate meaningful interpretations, for example language. The language function of the cortex extends beyond oral input to the comprehension of written language. The temporal lobe also has a role in memory. The occipital lobe receives inputs from the retina, the light-sensitive surface at the back of our eye, and processes these visual stimuli to generate meaningful interpretations that are passed to other lobes. Visual perception, for example, of objects, words, faces, occurs in and on the boundaries of the occipital cortex. The brain contains a large number of structures or areas that have specialist functions. It has a functional anatomy. Although the functional anatomy is very complex, there are well-defined correlations between specific types of thinking and behaviour and specific structures of the brain. In this section you can explore a limited selection of these structures. Later in the activity you will be able to explore how these and other areas may be involved in autism. The limbic system. The limbic system is a loosely defined set of structures on the inner ring of the cortex. The term limbic comes from the Latin word limbus, meaning border or edge. The limbic system includes the amygdala, the cingulate cortex, the thalamus, and the hippocampus, and according to some neuroscientists, the orbitofrontal cortex. It's involved in memory, decision making, and emotion, and has outputs on the level of the whole body through its influence on hormones and nervous system. The amygdala is part of the limbic system. It governs the emotional response to stimuli, for example, a facial expression for anger or fear. The cingulate gyrus is part of the limbic system also. It coordinates sensory input with emotions and is involved in regulating attention. Orbitofrontal cortex is sometimes thought of as being part of the limbic system. It is deeply involved in decision making. Damage to this part of the cortex leaves a person with their ability to reason intact, but with an inability to follow through into appropriate action. This could be very common for people with autism or people with autistic spectrum conditions. The fusiform gyrus is a key contributor to visual perception. Its capabilities are still to be understood completely, but it is well established that it has a central role in processing human faces. Accidental damage to this area, which could be caused by dementia or some other brain injury, is likely to lead to the inability to recognise faces, even of close family and friends. The cerebellum. Traditionally, the cerebellum, the structure that lies underneath the back of the cortex, has been associated with coordination and motor control. However, recent studies have shown its functions are far wider. It also appears to influence attention and the processing of sensory stimuli. The thalamus is part of the limbic system. It acts as the brain's relay station, processing stimuli and passing them on to cortical structures. The so-called thalamocortical circuits are central to the direction of attention and arousal. Damage to the thalamus can lead to an inability to respond to stimuli and even to a comatose state. The hippocampus is part of the limbic system and has a central role in long-term memory, learning and navigation skills. In Alzheimer's disease, degradation of the hippocampus results in loss of memory and inability to navigate. Development before birth. In humans, as in all other vertebrates, the brain emerges from a structure known as a neural tube. By 25 days after conception, the neural tube has developed 3 prominent swellings. At about 40 days, the tube begins to bend. This is necessary if the brain is to continue to grow within the restricted space enclosed by the emerging skull. By about 50 days, the cerebral hemispheres are becoming visible. By 100 days, they are very obvious. During the following months, they expand enormously and become the dominant features. The folds in the brain, the sulci and the gyri are formed by the 8th month, and the brain has its full complement of neurons, its processing cells, shortly after birth. The growth structure of the infant brain is nearly the same as that of the adult, but the brain continues to develop throughout life. During childhood, the brain will increase in weight by about a factor of 3 in the first 2 years of life, by a factor of 4 by early adulthood, and a subsequent slow decline in weight as we age. The number of neurons is fairly constant from shortly after birth to adulthood. The increase in weight is not due to an increase in the number of neurons. It is associated by 2 processes. The first is an increase in the number of different type of cells, glial cells. These do not process information themselves, but support the neurons physically and maintain the chemical environment that is necessary for neurons to function properly. The second is an increase in the number of connections formed between the neurons. The systems of the brain develop in sequence. Those necessary for survival, the sensory system develop rapidly. The baby must be able to feel, suck and swallow. Sight and hearing too develop rapidly. Language and motor skills are progressively developed from rapid maturation of the associated brain systems in the latter part of the first 3 years. Cognitive systems continue to develop alongside social skills. but it's not until well into the adolescence that the systems of decision making are fully present. Neurons and networks. Processing and transmission of signals in the brain are both carried out by neurons. These vary greatly in size and shape, but they all consist of three structures: an input set of fibers, the dendrites that receive signals, a cell body, and an output, the axon that carries a signal to the other cells. The functional anatomy of the brain is determined by the connections between the axons and the dendrites. These are called the synapses. Signals flow through the synapses from the axons to the dendrites. The neuron responds to these multiple inputs. With the right set of inputs, it fires, sending a pulse or action potential onto other neurons. These other neurons can in turn signal to other cells, resulting in information processing throughout the nervous system. In the first few years of life, the neurons in the cortex develop rapidly in complexity. This development is guided by signals from the genes and from the environment. As the synapses proliferate and strengthen selectively, they form the neural systems that enable us to carry out the whole range of human functions. This process of synaptic change, which goes on throughout life, is known as plasticity. The networks are built up, connect neurons locally and across the brain. The end result is staggering in its complexity. The adult brain, the average neuron is connected to 10 to 20,000 other neurons. The speed of the networks increases too, as electricity insulating glial cells form around the long axons that connect different parts of the cortex. This process is known as myelination. It is in the myelin that makes the white matter. The brain's development is governed by feedback. We interact with our surroundings. With repeated exposure, neural systems are activated. Networks that are used frequently are strengthened and refined. Networks that are not used simply disappear. Through this mechanism, the brain becomes more effective in processing information. Our understanding of the brain's networks is still very limited, but the functional imaging methods are beginning to allow us to see the networks and the activity within them.